area conference attendees. I'm Corey Gucker. I work with the Great Basin Fire Science Exchange. I'm currently working with Nancy Shaw in the development of an online book which synthesizes the current knowledge and practical experience related to the biology, ecology, and use of native forbs in restoration. The book is designed to aid seed collectors, seed growers, nurserymen, landowners, and land managers as they increase the supply and use of native forbs in restoration. The project is primarily funded by the BLM, with additional support from the Forest Service's Rocky Mountain Research Station, the Plant Conservation Alliance, Great Basin Native Plant Project, and the Great Basin Fire Science Exchange. Forbs, or wildflowers, are important parts of vegetation and animal communities. They grow in multiple forms, occupying above and below ground space, as well as available resources within plant communities. Following fires, it is typically forbs and grasses that recover first and occupy the site to the exclusion of non-native invasive species. Forbs are eaten by a diversity of wildlife species. They provide an abundance of invertebrate populations, which are especially important to birds. Forbs are also incredibly important to sustaining pollinator populations. Wildflowers also trigger the happy brain chemicals in humans and make great additions to low maintenance landscaping in urban areas, campgrounds, along roadsides, and the like. Although the importance of forbs to ecosystem structure, function, and resilience has long been recognized, the knowledge needed to be successful using forb species in restoration has trailed that of grasses and shrubs. While past research has provided information about the ecology of many forb species, there are many knowledge gaps that are needed to be addressed before there can be widespread use of forbs in restoration. Over the past 25 or so years, several land management agencies, researchers, laboratories, and industry folks have worked together to address the biology, seed technology, and practical knowledge needed to increase the supply and use of forbs in restoration. The agency and industry efforts have produced an abundance of information and guidance. However, this information is widely scattered. So this online book gathers and synthesizes all existing information in publications, reports, as well as data and personal input from seed labs, seed extractories, and seed collectors. The BLM provided us a list of 98 forb species occurring in the Great Basin and beyond. The list was prioritized based on the projected importance of each species for those Great Basin sites in the greatest need of restoration. To date, we have completed 25 species-focused chapters in the online book. The information gathering, summarizing, and reviewing process for each chapter takes from 2 to 5 months to complete. We also have plans to develop a table which will further summarize each of the chapter sections of each species, which could then be used as a quick reference when developing a seed mix or growing nursery stock. Each species-focused chapter is posted online as an interactive PDF as soon as it's finalized. The chapters are organized as you see here. General biology and ecology information is presented first, followed by sections that address seed technology, seed production, nursery production, and wildland restoration. So now I'm going to walk you through just a little bit what you can, close of what you can learn from the online forb book. The first 3 photos, the top and bottom left, are of parsnip flower buckwheat, where you can see it grows with sagebrush and other semi-desert shrubs in dry, rocky soils. The plants grow as low spreading clumps, suggesting this species would be useful for soil stabilization, even at a dry, rocky soil site. The photo in the lower right corner is Gray's lomatium. It's a mid to late cereal species. The plant pictured is 3 years old. Above ground growth of Gray's biscuit root is slow as plants devote initial resources and energy into taproot development. The species is relatively long-lived and recovers quickly following disturbances. So grazed biscuit root will not provide rapid soil cover once established, but should provide longevity and resilience to the restoration site. Species characteristics are not the only important factor in determining which species to add to your revegetation seed mix. In many cases, your restoration goals will aid in choosing which species to include. In this example, we feature fernleaf biscuit root, which is a long-lived mid to late cereal species that greens up very early in the growing season. It produces a large taproot, making it drought and disturbance tolerant. It provides important spring forage for native ungulates and livestock, and was found in the diets of both black bears and grizzly bears. Fernleaf biscuit root also provides a spring food source for pollinators and is a host plant for swallowtail butterfly species. Western Indian tribes harvested, ate, and preserved the taproots and used stems and roots to make inhalants, teas, and poultices to treat respiratory illnesses and skin wounds. Recent laboratory screenings revealed that fernleaf biscuit root has antiviral and antibacterial properties, and today is collected and sold as an herbal medicine. This information suggests fernleaf biscuit root would be useful in restoration plantings for pollinators and wildlife. However, it should be used in conjunction and strategically planted with early cereal, short-lived species that can provide cover and flowers to the restoration site initially. The revegetation mix should also include other mid to late season blooming forbs so that the site will provide for wildlife forage and support pollinators later in the growing season. As indicated earlier, the latter half of each species chapter focuses on obtaining the seed and plants to use in restoration. Pictured here is arrowleaf balsam root. For each chapter, a map like the one you see here is provided. The black dots are historical and contemporary, georeferenced herbarium specimens and observational data. In this case, the collections and observations range from 1870 to 2016. This distribution is displayed in relationship with generalized seed zones, the colored regions, and Omernick level 3 ecoregions, which are the black outlines. The chapter also provides links to interactive maps, legends, and mobile apps. Once appropriate seed zones are identified, plants can be monitored for the optimal timing for seed collection. For arrowleaf balsam root, seeds ripen relatively uniformly. Seed is ready for harvest when the inflorescence is dry, and seeds are dark brown to black. However, a high percentage of seed often fails to ripen or fill because of insect damage. Because filled and non-filled seeds appear nearly identical, seeds must be cut with a knife to determine fill. As flowers are maturing, collectors can cut across the flower head horizontally to see how many flowers are producing viable seeds, which will appear white and plump. This type of monitoring allows collectors to focus their efforts on those sites with plants that are producing a high percentage of filled seed. For many species, wildland seed cannot be collected in the abundance needed for restoration projects. And when we start using Forbs more in restoration, agricultural production of seed will be necessary. Pictured here is hoary tansy ester. It was grown in seed production plots by researchers at the NRCS Aberdeen Plant Materials Center and Oregon State University's Malheur Experiment Station. These trial plots provided information on site preparation, seeding, stand maintenance, and methods for maximizing seed yields. Hoary tansy ester is indeterminate. As you can see in the top left picture, this single hoary tansy ester plant is dispersing mature seed, but also has freshly opened flowers, which have not yet set seed. Hoary tansy ester produces a mature seed over a period of about 4 weeks, making large-scale harvests challenging. Researchers were challenged to come up with harvest techniques that could maximize seed yields in the agricultural setting, because initial seed harvests made by swathing and windrowing whole plants resulted in harvests that included an abundance of unripe seed and required extensive cleaning. However, by growing plants in weed barrier fabric, they were able to non-destructively and repeatedly harvest Torrey tansy ester by vacuuming the seed that dropped on the weed barrier fabric, as well as ripe seed from the plant. Vacuuming, although labor intensive, allowed for multiple harvests of only ripe seed with limited trash, and thus required less cleaning. Seed cleaning, seed storage, and seed testing are other topics that are addressed in each Forb book chapter. Finally, each chapter includes any and all information we find on nursery production of plants and any accounts of seeding or planting in wildland settings, roadsides, or in reclamation projects. For most chapters, the wildland seeding and planting section is often sparse, which highlights the need for knowledge to help ramp up the inclusion of Forbs in wildland revegetation and restoration. In many cases, we found very few field studies evaluating seeding or planting of native Forbs on wildland sites. So I want to thank you for your attention, and I really hope that you'll check out the online book. What we've provided here is really just the tip of the iceberg on what you can learn and find from the online book, Western Forbs, Biology, Ecology, and Use and Restoration. It is housed on the Great Basin Fire Science Exchange website that you see here. And if you have questions or comments, I welcome those, and you can contact me using the email or phone number you see here. Again, thank you for your time.